To begin talking about the endosymbiotic theory, we are firstly going to discuss 5 similarities between the mitochondria, chloroplast, and bacterial cells. The first similarity is that all 3 of them share is the ability to produce or release energy. The chloroplast does this by photosynthesis, the mitochondria by cellular respiration, and the bacterial cell by using its membrane. The second similarity they all share is that they all contain their own DNA. The third similarity that they share is that they are relatively small and similar in size. Another similarity is the presence of a phospholipid bilayer in all the membranes. Finally, the last similarity is the presence of fluids inside each membrane. The second topic that we are going to discuss today is the formation of a mutually beneficial relationship between an aerobic cell and an anaerobic cell. In order for us to continue, we must first define the 2 forms of energy sources. Anaerobic is the process of gaining energy without the use of oxygen. We can see this in the first step of cellular respiration with glycolysis, which generates 2 ATP. Aerobic is the process that requires oxygen. This can be seen in the electron transport chain and the Krebs cycle, where 36 ATP is produced. If a mutually beneficial relationship were to occur, then the benefit of the 2 processes working together would be that if one was to fail, the other would still function.
Explain why there are 2 lipid bilayers around mitochondria and chloroplasts. Describe how the double membrane allows for enhanced functions in metabolism. Mitochondria and chloroplasts have 2 lipid bilayers because the inner lipid bilayer would have been the bacterial cell's plasma membrane, while the outer lipid bilayer came from the cell that engulfed it. This supports the endosymbiotic theory that states that eukaryotes evolved from a process where different types of free-living prokaryotes became incorporated inside larger prokaryotic cells that would eventually form mitochondria and chloroplasts. The double membrane of mitochondria and chloroplasts allows for enhanced function and metabolism because the inner membrane that folds over many times creates a greater surface area for the cell. Therefore, the increased surface area allows for more chemical reactions to occur. Mitochondria breaks down nutrients, which is the process of ATP, and generates energy, while chloroplast is a photosynthetic organelle, and it converts energy from the sunlight to chemical energy. The double membrane in both of these organelles help them have more activity due to the increased surface area from the inner membrane.
The endosymbiotic theory was first proposed by Lynn Margulis in the 1960s. It explains how eukaryotic cells may have evolved from prokaryotic cells. She was doing research on the origin of eukaryotic cells. She looked at all the data about prokaryotes, eukaryotes, and organelles. She proposed that the similarities between prokaryotes and organelles, together with their appearance in the fossil record, could best be explained by endosymbiosis. Her hypothesis originally proposed that mitochondria are the results of endocytosis of aerobic bacteria. Chloroplasts are the results of endocytosis of photosynthetic bacteria, in both cases by large anaerobic bacteria who would not otherwise be able to exist in an aerobic environment. This arrangement became a mutually beneficial relationship for both cells. And the result was that a cell with a double membrane bound organelle, the inner lipid bilayer would have been the bacterial cell's plasma membrane, and the outer lipid bilayer came from the cell that engulfed it. The evidence is that if the organelles were really bacterial symbionts, they would have their own DNA. If her theory did best explain the origin of eukaryotic cells, she reasoned organelles would have DNA that resembled bacterial DNA and be different from the cell's DNA. We also have endosymbiotic theory, primary and secondary. Primary endosymbiosis occurs when a eukaryotic cell engulfs and absorbs a prokaryotic cell, such as a small cell that undergoes photosynthesis. Secondary endosymbiosis occurs when a eukaryotic cell engulfs and absorbs another eukaryotic cell. The 2 main differences between primary and secondary endosymbiosis is that the sequencing of the engulfment and absorbent of the genetic material, and whether prokaryotic or eukaryotic cells are absorbed by the eukaryotic cell.
The theory of endosymbiosis can have a grand impact on how one views society and how it functions. First of all, the human species as we know it could not exist without endosymbiosis. What would become the mitochondria were other microorganisms that were ingested by the cell through the process of endocytosis. The mitochondria greatly increased the efficiency of energy conversion within these cells. An increase in productivity led to multicellular organisms. Life, which began as a single cellular microorganism, would develop into the great diverse biosphere that we know today. With our knowledge of endosymbiosis, how can we use this to our advantage? One way would be to develop the field of genealogy, or the study of ancestry. For instance, since mitochondria used to be separate organisms, it has its own DNA. This DNA is unique since it is only inherited down the female line. Based on this knowledge, we could begin a more accurate system of tracking family trees. Endosymbiosis can also affect belief systems. Since all eukaryotes have a mitochondria or chloroplast, all multicellular life on Earth stem from one single-celled organism. This theory supports the concept of evolution, and that natural selection leads to differentiation between different species in different environments. This view opposes many religious institutions. For example, in Judeo-Christian culture, every organism on Earth was individually created by God. However, since only prokaryotes can ingest other microorganisms, this supports the idea that all life had evolved from a single cellular organism. Lastly, the theory of endosymbiosis may raise questions about the self. Symbiosis refers to a beneficial relationship between two organisms. This may then raise the existential question of who are you, or rather, what are you? The mitochondria used to be another organism, and now it lives in every single one of your cells. You could not survive without it. You can no longer see yourself as a single organism. Rather, you must see yourself as a system of many organisms that work together to make you who you are.